Everyone, myself K.B.N. Ali Basha, Department of Botany, K.B.N. College, Vijayawada. It is the useful to the BSc Honors, Botany, Zoology, Microbiology, Biotechnology, etc. The paper 1 is the essential of biology. The second unit is a essential of botany. First one is the classification of plant kingdom. The plant kingdom is a plantae. Kingdom plantae includes all the plants. They are eukaryotic, multicellular and autotrophic organisms. The plant cell contains a rigged cell wall. Plants have chloroplast and chlorophyll pigment which is required for photosynthesis. The plant kingdom has been classified into 5 subgroups. First one is the Thallophyta, Bryophyta, Pteridophyta, Gymnosperms, Angiosperms. Thallophyta. Thallophytes lack a well-differentiated body structure and the plant body is thallus-like. Thallophytes includes plants with primitive and simple body structure. The plant body is thallus. They may be filamentous, colonial, branched or unbranched. Examples include green algae, red algae and brown algae. And example Volvox, Ficus, Pyrogyra, Cara, Polysiphonia, Althrix and etc. Next one is the bryophyte. Bryophytes do not have vascular tissues. The plant body has root-like, stem-like and leaf-like structures. Bryophytes are terrestrial plants but are known as the amphibians of the plant kingdom. As they require water for a sexual reproduction, they are present in moist and shady places. Bryophyta include mosses, hornworts and liverworts. Example Marchantia, Funaria. Examples of Bryophyta: liverworts, Marchantia, Riccia, Pilia, hornworts and mosses. These are the examples of the bryophytes. Next one is the pteridophytes. Pteridophytes have a well-differentiated plant body in root, stem and leaves. They have a vascular system for the conduction of water and other substances. Some of the common example: Selaginella, Equigistium, Petris, etc. Gymnosperms. Gymnosperms have a well-differentiated plant body and vascular tissues. They bear naked seeds. Seeds are not enclosed within the fruit. Example of gymnosperms are Cycas, Pinus, Ephedra, etc. Angiosperms. Angiosperms are seed-bearing vascular plants with a well-differentiated plant body. The seeds of angiosperms are enclosed within the fruits. Angiosperms are widely distributed and vary greatly in size. Example, Ulfia is a small measuring about 0.1 cm and eucalyptus trees are around 100 meters tall. Angiosperms are further divided into monocotyledons and dicotyledons according to the number of cotyledons present in the seeds. Some of the common examples are mango, rose, tomato, onion, wheat, maize, etc. Next one is the cryptogams and phanerogams. The plant kingdom is classified into 2 groups. Cryptogams. There are the non-flowering and non-seed bearing plants. Example Thallophyta, Bryophyta, Pteridophyta. Phanerogams, flowering and seed bearing plants. Example Gymnosperms, Angiosperms. Next topic is the photosynthesis. Photosynthesis is a process by which phototrophs convert light energy into chemical energy which is later used to fuel cellular activities. The chemical energy is stored in the form of sugars which are created from the water and carbon dioxide. Photosynthesis definition stated that the process exclusively takes place in the chloroplast through photosynthetic pigments such as chlorophyll A, chlorophyll B, keratin and xanthophyll. All green plants and a few other autotrophic organisms utilize photosynthesis to synthesize nutrients by using carbon dioxide, water and sunlight. The byproduct of the photosynthesis process is the oxygen. Let us have a detailed look at the process, reaction and importance of the photosynthesis. The photosynthesis reaction involves 2 reactants, carbon dioxide and water. These 2 reactants yield 2 products, namely oxygen and glucose. Hence, the photosynthesis reaction is considered to be an endothermic reaction. 6CO2 plus 6H2O gives rise to C6H12O6 plus 6O2. The process of photosynthesis occurs in 2 stages: light dependent reaction or light reaction, light independent reaction or dark reaction. The importance of photosynthesis. The photosynthesis is essential for the existence of all life on earth. It serves a crucial role in the food chain. The plants create their food using this process, thereby forming the primary producers. Photosynthesis is also responsible for the production of the oxygen which is needed by the most organisms for their survival. The next one is the respiration. Respiration is a metabolic process that occurs in all organisms. It is a biochemical process that occurs within the cell of organisms. In this process, the energy is produced by the breakdown of glucose, which is further used by cells to perform various functions. Every living organism, from a single-celled organism to dominant to multicellular organisms, perform the respiration. The types of respiration. There are two types of respiration: aerobic respiration and anaerobic respiration. The aerobic respiration, it is type of cellular respiration that takes place in the presence of oxygen to produce energy. It is a continuous process that takes place within the cell of animal and plants. This process can be explained with the help of the chemical reaction C6H12O6, it is the glucose, the 6CO2, it is the oxygen, gives rise to carbon dioxide 6CO2 plus water 6H2O plus energy ATP. Anaerobic respiration. It is a type of cellular respiration that takes place in the absence of oxygen to produce the energy. The chemical equation for the anaerobic respiration is glucose C6H12O6 gives rise to alcohol 2C2H5OH plus carbon dioxide 2CO2 plus energy ATP. The three phases of respirations are: first one, the glycolysis. The molecule of glucose get converted into pyruvic acid, which is oxidized to carbon dioxide and water, leaving two carbon molecules known as acetyl coenzyme A. During the process of glycolysis, two molecules of ATP and NADPH are produced. Pyruvate enters the inner matrix of the mitochondria and undergoes oxidation in the Krebs cycle. The second one is the oxidative phosphorylation. Oxidative phosphorylation is the process in which ATP molecules are formed as a result of the transfer of electron from NADH or FADH to O2 by a series of electron carriers. This process takes place within the mitochondria of a cell. Third one is the citric acid cycle. This is also known as the tricarboxylic acid cycle or Krebs cycle. Two ATP molecules are produced in the each phase of the citric acid cycle, and it takes place within the mitochondrial matrix of a cell. The electrons generated in Krebs cycle move across the mitochondrial matrix. The next topic is the transpiration in plants. Like all living organisms, plants also require an excretory system to discharge excess water from their body. This process of elimination of excess water from the plant body is known as transpiration. It is generally the evaporation of water from the surface of the leaves. During the process of transpiration, water molecules in the plant's tissues are removed from the aerial part of the plants. Only a small amount of water absorbed by the plants is utilized in growth and development. The rest is eliminated in the form of the transpiration. Types of transpiration. There are three different of transpiration in plants. Stomatal transpiration. It is the evaporation of the water from the stomata of these plants. Most of the water from the plants is transferred this way. The water near the surface of the leaves changes into vapor and evaporates when the stomata are open. Lenticular transpiration. Lenticular or mint opening in the bark of a branches and twigs. Evaporation of water from the lenticels of the plants is known as a lenticular transpiration. Now, lenticels are not present in all the plants. A minimal amount of water is lost through the lenticels. Next one is the cuticular transpiration. It is the evaporation of water from the cuticle of the plants. The cuticle is a waxy covering on the surface of the leaves of the plants. About 5 to 10% of the water from the leaves is lost through the cuticular transpiration. During dry condition, when the stomata are closed, more water is transferred through the cuticles. The next one is the plant growth regulators. The plants require light, water, oxygen and minerals and other nutrients for their growth and development. Apart from this external requirement, plants also depend upon the certain organic components to signal, regulate and control the growth of the plant. These are the collectively called plant growth regulators or plant growth hormones. The types of plant growth regulators. The plant growth hormones or regulators are the following types. Plant growth promoters, auxins, gibberellins, cytokinins. The plant growth inhibitor, abscisic acid and ethylene. Next one is the auxin. The first phytohormone to be discovered is the oxygen, and it was discovered by the biologist Charles Darwin. Auxins are one of the most important plant hormones. The chiefly naturally occurring auxin is the indole-3 acetic acid, IAA, and other related compounds. The term oxygen is derived from the Greek language meaning to the grow. Next one, the gibberellins. Gibberellins are an extensive chemical family based on the end gibberellin structure. The first gibberellin to be discovered was the gibberellic acid. Now there are more than 100 types of gibberellins and are mainly gathered from the variety of organisms from fungi to higher plants. Cytokinins. These are produced in the regions where cell division occurs, mostly in the roots and shoots. They help in the production of a new leaves, lateral shoot growth, chloroplast in leaves, etc. They help in the overcoming apical dominance and delay aging of the leaves. Abscisic acid. It is a growth inhibitor which was discovered in the 1960s. It was initially called dormant. Later, another compound, abscission 2, was discovered and are commonly called as the abscisic acid. This growth inhibitor is synthesized within the stem, leaves, fruits, and seeds of the plants. Ethylene. Ethylene is a simple gaseous plant growth regulator synthesized by the most of the plant organ, includes ripening of fruits and aging the tissues. It is an unstructured hydrocarbon having a double covalent bond between and adjacent to carbon atoms. The next topic is the flower of structure. In the flowering plants, reproductive structures are present called flowers. The major part of the flower are sepals, petals, stamen, and carpels. The sepals are calyx. Sepals are the green color portion beneath of the petals. Sepals help in the protecting the rising buds. Next one is the petal or corolla. The petals are the bright colored part of the flower that attract to bees, insects, and birds, in which color may vary from plant to plant. The stamens are androecium. The male reproductive part are known as the stamens or androecium, which is produce pollen grains. Two parts of the androecium are anther and filament. Carpel or gynoecium. Female reproductive organs are called pistils or carpels. These are the innermost parts where seed formation takes place. It consists of three parts: stigma, style, and ovary. Stigma, swollen receptive portion of the pistil. Style, long tube-like structure connecting the ovary and the stigma. Ovary, but bottommost region where ovules are present and seed formation takes place. The next topic is microsporogenesis. Microsporogenesis is the formation of microspores inside the microsporangium by meiotic or reduction division. The formation of pollen grains from the microspores is termed the microgametogenesis. The formation of microspores, the developing anther consists of two types of cells, reproductive and non-reproductive cells. The diploid cells are the reproductive cells in the sporogenous tissue of the microsporangium called myocytes or pollen mother cells divide by meiosis to produce the 4 haploid microspores. The first reduction division gives to 2 nuclei and the second division finally gives to a tetra of microspores. The non-reproductive cells form the anther tissue layers including epidermal, tapetal, and cortical layers. The tapetal layer functions are a nutritive tissue for the development of anther. Formation of pollen grains. The formation of pollen grains from the microspores is termed as a microgametogenesis. It begins with expansion of the microspores and the formation of a large vacuole. It is followed by the displacement of the microspore nucleus from the center to the one side of the cell against the microspore wall. Here, the first pollen mitosis takes place that produces a large vegetative cell and a small generative cell, the cell containing the haploid nucleus. The regenerative cell that was attached to the microspore wall starts detaching, and it is engulfed by the vegetative cell, forming the unique cell within the cell structure. The engulfed vegetative cells then undergo pollen meiosis to form a 2 pollen sperm cells packed in the cytoplasm. This is the 2-stage formation of pollen grains from the microsporogenesis and microsporogenesis and microgametogenesis. Macrosporogenesis is the next topic. The process of the development of megaspores from the megaspore mother cells is known as a megasporogenesis. The megaspores are developed into the female reproductive organs. Development. A layer of sporogenous tissue is present in the ovules of the female flowering plant. The megaspore mother cell MMC are developed from the sporogenous tissue. It is a large diploid cell with a prominent nucleus and a dense cytoplasm. The MMC undergoes meiotic nucleus division to produce the 4 haploid megaspores. The megaspores are arranged axially called the linear tetrad. Out of the 4, only one megaspore is functional and the rest 3 degenerates. The megaspore at the collegial end is functional and the 3 towards micropylar degenerated. The one functional megaspore divides metatically to form a embryo sac, a process called megagametogenesis. It divides multiple times to produce the 8 haploid nuclei for the embryo sac, which is also called as a megagametophyte. The next topic is the pollination. Pollination is a method where pollen grains are picked from the anther, which is the male part of the flower, and the transferred to the flower's female part called the stigma. To make the pollination work successfully, the pollen grain must be transferred from the same species of flower. Types of pollination. All plants having flowers completely rely on pollination method for the reproduction. There are 2 types of pollination: self-pollination and cross-pollination. Self-pollination occurs when the pollen grains fall directly from the anther into the stigma of the flower. Cross-pollination, it refers to the complex type of pollination that allows the transfer of pollen grains from the anther of the flower into the stigma of the anther flower. The next one is the fertilization. The fertilization can be defined as the fusion of the male gametes with the female gametes to form a diploid zygote. It is a physiochemical process which occurs after the pollination of the carpel. Types of fertilization. Porogamy is the common type of fertilization carried out in all angiosperms or flowering plants. In this type of fertilization, the pollen tube enters the ovule through the micropyle. Chalajagamy. This type of fertilization is carried out on all Casuarina species of plants. In this condition, the pollen tube enters the ovule through the pollen tube. Mesogamy. This type of fertilization is seen in all cucurbita plants such as a pumpkin, ridge gourd, bitter gourd and other gourd plants. In this type of fertilization, the pollen tube enters into ovule through the middle part or through the integuments of the ovule. Structure of monocot embryo. The monocot embryo is an embryo with the presence of only one cotyledon. In the context of the grass family, like a Gramineae, this is referred to as a scutellum. The single cotyledon is typically present at the lateral side of the embryonal axis. At the lower end of the radicle and a root cap are present and they are enclosed in the sheath called a coleorhiza. Structure of dicot embryo. The dicot embryo is the embryo of dicot plants that contains 2 cotyledons. Hence, these 2 cotyledons are present at the either side of the embryonal axis. Furthermore, these 2 cotyledons are located laterally. Here, epicotyle, which is the portion above the cotyledons, terminates with the pulmule, that is the further shoot. The pulmule is present distally in the embryo. The hypocotyl is the region that is the present below the level of the cotyledons. Finally, the hypocotyl terminates with the development of the embryo into a structure known as the radicle, that is the future root. The radicle is the root tip covered by the root cap known as the calyptra. Next one is the mushroom cultivation. There are 3 sorts of the mushrooms that are being cultivated in India. They are button mushroom, straw mushroom and oyster mushroom. Paddy straw mushrooms can be developed in temperatures ranging from the 35 degrees centigrade to 40 degrees centigrade. The major steps in the cultivation of common edible mushrooms. The first one is the composting. Composting involves the mixing of various components such as a wheat or paddy straw, chicken manure and organic and inorganic fertilizers in a fixed proportion. The temperature of the compost is maintained at the around 50 degrees centigrade. The compost is kept undisturbed for about 1 week. Second one is spanning. Mushroom seed in the form of the mycelium of mushroom to be grown introduced into the heap of compost and left for the spreading for around 2 days. Causing. Causing is the most important step of mushroom cultivation. It involves the spreading of a thin layer of soil over the compost. This provides humidity and support to the mushroom. It also serves to prevent the dissection of the compost heap and helps in the temperature regulation at around 20 degrees centigrade to 25 degrees centigrade to forbid the growth of the pest and diseases. The provision for the circulating air around the compost bed should also be made. Cropping and harvesting. Three major growth stages are observed before the mushroom attain the fully grown form. Firstly, the mycelium, a network of fibrous mass spread out the 2 to 6 weeks, followed by the tiny pin head stage and finally the button stage, which is marked by the increase in the mushroom size until it acquires the marketable size. Next one, the preservation. Mushrooms have a very short shelf life. Process such as a vacuum cooling, bombardment by gamma radiation, followed by the storage at 15 degrees centigrade freezing, drying in the solution of citric acid, ascorbic acid and brine, etc. are used for the preservation of mushrooms. Next one is the floriculture. Floriculture is the cultivation and trade of flowers. It encompasses flower production, processing, marketing and distribution. The first recorded evidence of the floriculture dates back to the early 1500 years when flowers were grown in the greenhouse in the Holland. Today, floriculture is the multi-billion-dollar industry with a global reach. In the United States, floriculture is nearly $20 billion industry, with California being the top producer of the flowers. The Netherlands is the world's largest exporter of the flowers, followed by the Germany and Italy. Types of floriculture. The below mentioned the types of floriculture. The bedding plants. These are annual plants that have been grown for the use in flowers bed outside. It begins with the sowing of seeds indoors during the off season and the transplanting of the plants later in the growing season. Hanging plants. These are the hanging basket plants, either annual or perennial, flowering of the foliage rows in decorative pots and suspended by the elegant rope from the ceiling in the patio, in the doorway section, or from the decorative plant poles. Potted plants. These plants are suitable for the indoor use as well as home and small garden. They are mainly foliage plants grow in pots, though some flowering plants are often used the potted plants. Dried plants. These are the flowers that are used after being dried for the various occasions. These flowers are dried using various preservatives. Next one is the cut flowers. Cut flowers are blooming flowers or flower buds that have been cut together with roots, branches and leaves for use in banquet or decorations. There are two types of cut flowers, a new cut flowers and non-fresh cut flowers, such as a preserved flowers, roses, carnation, orchids, chrysanthemums, lilies, gerbera and other fresh cut flowers are popular. Next one is the landscaping. The importance of landscaping is now realized by everyone in the world. The gardens serve the purpose of a public reaction by refreshing the body and the mind. They are virtually a retreat for the public from the harsh strains and stresses of a public life. Landscaping is done with a view of create a natural sense by the planting of a lawn, trees and shrubs. It is the imitation of the nature in the garden and improves the total living environment of the people. We live in the vast planet which is characterized by different types of landscaping depending upon the prevailing geographical and agroclimatic conditions. First, there are mountains, hills, glens, valleys, seas, rivers, forests, plains, deserts, lakes, swamps, streams, etc., which comprises major part of the natural landscape. The basic principles of the landscaping. Before actually drawing the master plan, the following points should be kept in mind in the home landscaping. First one is the background. The background in the garden, whether a wall, a tall trees or a hedge, should be a neutral in nature. That is to say this should not become a discreting the feature over the main feature of the garden. Contrast. The design should be such that they should be break monotony. To achieve this, a variations in from form, texture or color has to be brought in. Balance or proportion. A balance has been maintained between the different components, masses, forms, colors, etc. of a garden. In a formal garden, this is achieved by the balancing the quantity or by adjusting the various in an informal garden. This can be achieved by planting a small mass of color in front of a large neutral masses. Overcrowding of a plants or other garden features should be avoided. Next one is the open center. This central area of the garden should be left out any items of the major interest. The best way to achieve this is to have a lawn which also gives an effect of a largeness to the property. A specimen shrubs in the center of the lawn is the unsuitable as this counters the principles of the species, but a tree branching at a higher level from the ground could be planted. Repetition. The repetition or duplicating the some feature of the garden helps achieve the rhythm, balance and the unity. In a formal garden, generally the same feature is repeated, but for an informal design this need not to be so. Here one may repeat the color tone without a disturbing the texture, form and quantity. If there is the circular path, this can be repeated by the having 2 or 3 consecutive circular shaped beds of annual flowers, heads and shapes border. Thus, though shape is repeated, the variation in the texture, color and form ensure that the design does not look monotonous. Rhythm. A landscape designing should have an artistic sense to understand how to bring a rhythm in the design. Arranging the different elements haphazardly without harmony does not enhance beauty. Harmonious lines, often artistically curved, bring a rhythm to landscape. A group of shapes in front of a rockery breaks the rhythm. The repetition of certain elements such as a form enhance the rhythm. Variety. To break the monotony in the garden, variety is essential. This is the achieved by contrast of a color, form and texture. Planting of different seasonal flowers all in red color does not necessarily mean a variety. Thank you.